lecture on the caves that are in France and Spain, where there are prehistoric paintings of marvelous paintings of all kinds of animals, bison and deer and all kinds of things, from about 17,000 years ago and before. The people who painted them went to considerable effort to do that. They had to go deep underground, pitch black, no electricity, at great effort to carefully paint these really magnificent pieces. And that's not the first time that there's evidence that human culture has been fascinated with animals. And it continues. The ancient Maya in Mesoamerica have all kinds of paintings and sculpture of animals. And the story of the Popol Vuh, stories of where animals come from. Obviously, we're familiar with that in our own religious traditions. So there's been a long-term definition, in part, of what it means to be human by how we understand animals and where they come from. So that's only one reason I'm delighted that Tom Jackman from our biology department is willing to discuss this long-held question that humans seem to have of where did this riotous display of animals and types of come from, and how do we understand them? Who's gotten his plea and has been a member of our department now since '99, is working on and has worked on to form a history of the development of these forms, which fits in very nicely with where we've come so far with the previous discussions here in our series. So thanks so much for your presentation.
Sure.
Thanks for coming.
All right, so the title that was suggested to me, I decided to take it. I didn't change it. Is where did animals come from? And I think that the question implies, it doesn't necessarily mean, but it implies a common misunderstanding about evolution. I just wanted to talk about that a little bit. And it comes from Aristotle. Aristotle's Scala Natura, sometimes called the ladder of life or the great chain of life. And this sort of metaphor for life is sort of a common way that sort of non-evolutionary biologists think about evolution. So I want to talk about that. I want to talk about, instead of a ladder of life, a tree of life. Talk about life in terms of a tree. And then I want to really answer this question, where did animals come from? The answer might be, I wanted to address it seriously as a biologist, that is, where did animals come from? And the answer might be a little less interesting than you might think, but I want to address it and give you the as precise an answer as I can based on what we know now. And this involves research that has just been published this year, a lot of this stuff, the answer to where did animals come from. So that's an interesting thing. I'm going to talk about epistoconts. Anybody know what an epistocont is? No, I didn't think so. We are epistoconts, and the fungus in some of our socks are also epistoconts. Okay? Just to let you know. So that's epistoconts are the common ancestor of animals and fungi, okay? And all of the descendants that arose from them. Okay, so I'll talk about epistoconts. Because that's part. And then I figured that if you're not satisfied with where animals came from, I might just discuss about the evolution of animals overall. And so talk about major events in animal evolution, sort of if I had to pick the top 3 events in animal evolution, what would they be? And I'll tell you what they are. I also want to talk about sort of the evolution of the vertebrate eye as an example of how evolution is not exactly perfect. And I picked the eye because this is one that creationists often pick to say that evolution couldn't happen because look how perfect the eye is. Well, the vertebrate eye, at least, it's not, in some respects, is not perfect. And in fact, cephalopod, that is squid and octopus eyes, are actually, from a design standpoint, appear to be a better design. So I want to talk about that and what it means, and sort of the lesson it tells you in terms of evolution for the vertebrate eye. Then I want to talk sort of, I think when we talk about animals, I think usually when you ask the general public what an animal is, they'll come up with answers of mammals, which is just a group of animals. But most of the animals that we care about a lot are mammals. And mammals are also a very good story in evolutionary biology. In the last 20 years or so, and even especially in the last 10 years, the story of mammal evolution has really solidified. And there's a great combination of molecular data, fossil evidence, and DNA data, and morphological data, that have sort of come together to form this coherent story about the evolution of mammals, and it's a good one, so I want to tell you about that. And then I also, if I have time, talk about the evolution of whales as another sort of particularly good case study in the evolution of mammals. Okay, so let's get started here with where did animals come from. I got this from the humanthermodynamics.com. I just wanted to find an image on the internet that there's nothing inaccurate about this. This is the history of life on Earth. But it sort of points to this misunderstanding that I want to get at, that has to do with the great chain of being, this scala natura that Aristotle had proposed a long time ago and sort of has cemented in human thinking. And that's because we start off here with the origin of life on Earth about 3.5 billion years ago or so. And then we've got this long period of a few billion years with just single-celled organisms, or maybe cyanobacteria, sort of crude multicellular organisms. Not until about 540 million years ago that we have this explosion of multicellular life, sort of interesting life as we know it. And the end result here is drawn as sort of a human right here, and that's sort of the misconception I want to address. And that here is the. I just want to talk about why Aristotle was wrong about biology, at least some of biology. Aristotle did some really interesting stuff, including his observations of hyenas. Female hyenas have penises, and Aristotle noticed this, and so that was an important biological observation. But one of the things he proposed was that this ladder of life, which also included non-living organisms, so this might be a logical progression in the ladder of life. You have dirt, and then plants, and then worms above that, and lions above that, and humans above that. And this medieval representation of Aristotle's ladder of life sort of goes beyond where Aristotle talked about. So you can go above humans, and then you can go to angels and God. So you can extend it up. It's this idea that there's this progression from the simplest to the most complicated, and that either we're the pinnacle as Aristotle saw it, or God, or some higher being is the pinnacle of this. And at least in biology, this is and the extension of that in terms of the great chain of being would be this sort of view of evolution that we go from a bacteria, and then we go to a worm, and then we go to a salamander, and then we go to a monkey, and then we go to a human. And that's what the study of evolution is for a lot of non-biologists. Evolution is trying to trace back from humans, go all the way back in time to this single-celled organism. But it's not the case, and there's sort of two major problems with this way of thinking, this scala natura, this ladder of life, this great chain of being. And so, if I can get this to work. Okay, there's two big problems with this great chain of being. The first one is that life is a tree, not a ladder. So the tree is a much better metaphor, and in fact, evolutionary biologists really are, one of the main goals of evolutionary biology is to reconstruct this tree of life. And the structure of the tree and not a ladder has important implications in terms of how we think about how evolution works, and in terms of how we think about humans. And it turns out humans are just one of the thousands of twigs on this tree. Not that humans are unimportant, and not that humans haven't achieved things that other animals haven't. I'm not saying that humans are the pinnacle in terms of consciousness and in terms of their brain and in terms of a lot of different things, but there's a lot of things that humans aren't the best at. So that this metaphor of a tree and not a ladder is an important one to consider. So we sort of have to abandon this idea of this ladder of life. And the second point, I'll get to this later on, but if we're interested in ancestors, sort of this great chain of being says that there's this focus on being able to trace ancestors all the way back to the origin of life on Earth, we're in trouble because 95% of all the species that ever exist, or more than that, it's 99.99% of all the species that ever existed are now extinct. And when we look at a tree with extinction on it, which I'll show you, we're unlikely to find ancestors in most cases, even when we have a pretty good fossil record. And even if we do find them, we're not sure which ones are the ancestors and which ones aren't. Okay? So we're kind of stuck with that. I'm going to show that at the end. And so I would posit that the focus really should be on the tree of life, reconstructing the tree of life accurately, and we can find out all kinds of interesting things from that. So let's get back to this question, where did animals come from? This is a tree. You can download this tree as a PDF. It's from David Hillis's lab at the University of Texas at Austin. This tree is for 3,000 species, sort of a sampling all of the species of life. It's a great underrepresentation of life on Earth. There's at least 3 million species, so this is 3,000 species. So there's a lot of things missing on this tree. So this is based on ribosomal RNA sequences. And again, you can download it, I think at full size, it's 54 inches or something like that. And I haven't printed it out, but because I don't didn't want to spend the money for the printer, but you can zoom in and out of this tree. And here's the animals right here. I've highlighted the animals here in pink. And you can see these other sort of major groups of organisms on this tree. And if you can't read this, this says "you are here" right here. Okay? So let's zoom on. You see, "you are here," and you still can't read the names yet. So the names are there on the tree. These are the names of the species. So here we're within animals, and we can zoom in a little closer, and maybe somebody could read this, but not quite. Let's see, we can have another. I've got it zoomed in even more. So that's about as much. So you can see here's Homo sapiens right here, and in fact, if you know any of these names here, here's Homo sapiens, here's a mouse, here's a rat, and here's a rabbit. Those are the only 4 mammals they sampled for this. So mammals are a little bit undersampled in this tree. It's partly because in 2003, the phylogenetic relationships of mammals were already pretty well understood. Okay, and so they tried to take a sampling of species for things we don't understand. And so we've only got 4 mammals on this tree, but it's not a huge underrepresentation given the fact that this is 3,000 species out of potential millions of species that exist on Earth today. Okay, so what about if we go back to all the animals? So we're in the animals here. These are all different animals. Where did animals come from? And now I'm highlighting. I twisted the tree around so maybe you can see. You can see that the fungi are here. And the animals right here. Here's Homo sapiens right here on the new branch of the tree there. We've got, and if you go back to the node in the tree where animals are, the closest relative of animals are fungi. Okay? So that's where this name came from. When they started looking at these DNA sequences, they realized that the closest relative of animals are fungi. All right? And so they came up with this name. The name is opisthecons. So the opisthecons are the clade that includes the common ancestor of animals and fungi. And if we want to know where animals came from, we have to go back about 1 billion years ago, maybe a little more, to the common ancestor of fungi and animals. And it was most certainly a unicellular organism that existed in the ocean somewhere, and nothing like it lives today. Okay, so I think that there really is no organism we can point to and say this is the common ancestor of fungi and animals. There are a number of single-celled organisms, however, that are very strange, and we only have recently come to realize that they're actually part of this opisthecon clade. And I want to talk about that. So this is from Trends in Genetics in 2007. Okay, this is a paper called "The Origins of Multicellularity." Right, so fungi, or at least many fungi are multicellular. So you have mushrooms and other things that are multicellular. And animals, nearly all animals are multicellular. And so this is a initiative to be able to understand how these 2 lineages independently obtained multicellularity, the fungi and then animals. So again, about 1 billion years ago, the unicellular common ancestor of all animals. So right here, here's the unicellular common ancestor of animals. It's right there. There's metazoa, so those are all animals. I probably should have mentioned this before, but when you look at animal diversity, you might ask, well, what are animals and what aren't animals? So I'll just name a couple of things that are maybe the least animal-like of the animals that we know to be animals. And one are sponges. Okay, so that's the first group that divides off in our phylogenetic tree of animals. Are sponges. They're the earliest division, but they are animals. And so are corals. If you look at corals, are animals. Okay, so these are radially symmetrical animals. They don't have a head, but they do qualify as animals. So those are some of the sort of least animal-like of all of the animals we might think about. But you'll notice here's this group of organisms, which when I saw this, I didn't know what they were. I had heard of these before. I'd heard of these chenoflagellates before, but I hadn't heard of these, and that's because some of these, like this one was discovered in 2006. So that's why I never heard of that one. These things, these are called ichthyosporia. These infect fish, and they thought they were fungi. They're not fungi. They're not chitin-based, actually. But so here are these are protists. So if we look at the different animal kingdoms, we have animal, fungi, plants, and then we've got these protists, which are this grab bag of these single-celled organisms, which are all weird. They're very bizarre in what they do. Their DNA is weird, and so there's sort of this odd collection. So here we've got included in this clade, this opisthecons, are now these protists, which appear to be the closest relatives of animals. And although it doesn't help us all that much, because in terms of figuring out what do you think this unicellular common ancestor looked like. And that's because we know that if we look at the tree of life and we look at these as living organisms today, these things are really different from each other, and they're really different from modern animals. So there's no reason to expect that the ancestor was anything like any of these things that are living today. I mean, maybe they are, maybe like one of these things, but these things are all pretty different from each other. So I just thought I'd show you a couple examples of these protists, these single-celled organisms, which we only recently now know to be the closest relatives of animals. Here's canaflagellates, and they're not actually as exciting as they look here. They've been dyed with, they've been stained with like fluorescent dyes, so they look interesting. So they're these single-celled organisms. They have a flagellum here, and they have this collar. All of them have this sort of collar-like structure here. And so here's sort of the morphology of a canaflagellate. They have a complete genome now of one of these canaflagellates, which sort of is a key factor for figuring out this evolution of whole genomes. I mean, the evolution of multicellularity that have evolved maybe in fungi and these things independently. So, canaflagellates, and here's another example. This is from this Trend in Genetics article. This is Spheroforma arctica, which form these semi-multicellular clusters, I guess, what you call them. What do they call them? They call them pseudo-multicellular colonies. So they're sort of multicellular, maybe. And there's really not that many images of them because they're not terribly interesting looking. And they're not that well known. They don't know that much about the biology of these things. So there's a lot and a lot of this is very recent. Like I said, some of these things were just discovered in 2006. So if we want to, so let's just go back. I want to review. Where did animals come from? They came from the ocean about 1 billion years ago, and they were unicellular, and they're unlike anything that's living today. I'm pretty confident of that, just because these things are really diverse, living in these specialized habitats, like inside of a fish, or in particular places that we don't think that that's where the common ancestor of all animals was living. So that's where animals come from. They come from a unicellular ancestor about 1 billion years ago, but the interesting stuff that happened in terms of animal history begins about 540 million years ago. So these are my top 3 major events in animal history. I had to pick the top 3 events. If you had to describe everything that's ever happened in terms of animals, it's got to start at 540 million years ago. And what happens there is, so we've got this, we have to go back 1 billion years before we find an ancestor that animals share in common with any other organism. And we have to wait for almost 500 million years before anything interesting morphologically appears in the fossil record. Okay? And at that point, nearly all of the major groups of animals, that is the phyla of animals, appear all at once in the fossil record, with the exception of sponges and jellyfish I just talked about. So you get sponges and jellyfish a little bit before this point. You get some of those, but almost everything, all the phyla, except 2 phyla, I guess, but most of the phyla all appear at once in the fossil record of 540 million years ago. So that's got to be a major event. And this is called the Cambrian explosion. And one of the great unanswered questions still to this day, and I'll show you some of the most recent research, does this appearance in the fossil record, does that reflect a burst of speciation between all of these different organisms in a very short period of time, or is it an artifact of the fossils? That is, the oceans weren't, and there was something wrong about the way fossils are preserved, and it wasn't until this point that we actually got the good fossils that we can, right? Maybe these things all existed for a long period as multicellular organisms before that, but we just don't have the fossils, right? So there's still this controversy whether or not this Cambrian explosion represents a real explosion in terms of a whole bunch of animals just diversifying in a rapid and short period of time into all of the forms that we see today, or is it an artifact of the fossils? Okay, so I want to address that. It's still, I'd say this is still somewhat of an unanswered question in evolutionary biology. The second one, I won't talk about that much. It's very interesting, but I'm not going to spend any time on it, is the end Permian extinction. This is the largest extinction ever on the Earth as far as we know it. 251 million years ago, 95%, and this has been updated in recent times, that they think 95% of all marine species at least go extinct. That's where our best fossil records are of that time. So we've got this huge extinction event occurring 250 million years ago. It's not as simple an extinction event as the next one I'm going to talk about in some detail, but the causes of it are being sort of uncovered in a more and more. So it involves a complex combination of meteors and other factors that made everything go extinct in a very short period of time. But the one that most people are familiar with in terms of animal history, and this may be more famous than important, but it's certainly important in terms of mammal evolution, which I'm going to talk about, and that's the end of the Cretaceous. So if we look at all animals and plants, it turns out there's some groups, we still don't know why, that were not as affected as dinosaurs were by this event. So 65 million years ago, and now the evidence for this is very strong, that a big, huge meteor hit the Yucatan Peninsula and caused a gigantic tidal wave, which wiped out everything around there and probably changed the atmosphere of the Earth enough to wipe out all of the dinosaurs. Okay? So the end of the Cretaceous 65 million years ago wipes out the dinosaurs, and the traditional story that's been told is that this opened up the door for mammals. Right? You've got rats or something like that living in trees and hiding out. They're all small, and then the meteor hits, the dinosaurs go extinct, and then they go crazy, and you get all the diversity of mammals. So that story's been questioned too. It's been revised, and I'll talk a little bit about that. Although what's still true is the dinosaurs were wiped out, and mammals did diversify, at least in numbers of species at that point. But the story's a little bit different from that. All right. So I'm going to go back to this point, this Cambrian explosion, and what does our modern data tell us about, our modern molecular data, that is DNA sequence data, tell us about this Cambrian explosion? Is it a real event involving a burst of speciation, or is it an artifact of the fossils because something happened in terms of the fossil preservation that we don't see any fossils before this point? And so we can contrast these hypotheses in terms of a phylogenetic tree this way. So this is from Science in 2005. This is a paper. This is the sort of review of the paper that appeared, sort of the news and views. Is the Big Bang in animal evolution real? All right. So this is this would be the animal Big Bang is the Cambrian explosion. And so here we've got the Cambrian right here, which is denoted by a C at 543 million years ago. The black lines indicate we've got fossils. So we've got sponges and jellyfish, which go before the Cambrian explosion. But then we've got all these other major groups, and there's even more of them. But these are the ones they sampled for this huge study. Okay, and we've got sponges, jellyfishes, and corals. Here we're chordates, things with backbones. We got flatworms, no fossils of flatworms. Nematodes, another kind of worm. Mollusks that include the clams and squid and snails. Annelids, which include leeches and earthworms. And we've got arthropods and this enigmatic Priapulida phylum that's also been included in this study. There's a lot of phyla that weren't sampled, but the reason what they did in this study is they actually sampled 50 different genes. So it's a huge data set. Most phylogenetic studies, so the ones that I do, maybe involve one gene, might involve 5 genes. But this one, they took 50 genes for a large number of these to try to get a definitive answer. And they didn't get a definitive answer, which suggested that this hypothesis, you can see that there's black lines right here. This is the hypothesis that the Cambrian explosion is a real reflection of the phylogenetic tree. There's this rapid burst early on of speciation that produced all these animal phyla in this relatively short period of time. The alternative hypothesis is represented by these red dashed lines, that these fossils just weren't preserved until this point, right? But the tree actually would extend a lot further back, and we'd have the tree well resolved. So if we look at the results from this 2005 paper, so this is the Science paper, and this is more of the sampling. But they sampled 50 different genes for this. And what's interesting about this is they also sampled the fungi. Remember the sister group, the other epistheconts. So we've got the fungi here, and when they sampled it, there's these numbers on the tree. As you go to 100, that means that you have more and more confidence in the phylogenetic tree. And they basically got very good support for this fungal tree of life. They're able to accurately reconstruct the fungal tree of life. And that's true on a fairly, not just for this study, but lots of studies have come up with a very nice tree of life for the fungi, for this sister group to us. But they didn't get these numbers here, which could approach 100. Many of them are below 100, and some of them, there's no support at all. So this is based on 50 different genes. So this is one of the reasons they said, well, here the fungi have this really nice tree based on these 50 genes, but the animals, not so good. So at least they suggested in this paper that it might support this rapid explosion. They also had some other hypotheses that I won't discuss for some of the reasons. One of them might be, even though they did 50 genes and they had all these animals, at least my impression was, we need to sample more. There's a lot more animals. Okay, it's not an easy thing to do to sample 50 genes from this many animals, but it turns out that it's been done. So this is sort of one side of the argument that Cambrian explosion 540 million years ago is a real expression of this rapid diversification. It's supported by this study that showed that the fungi get this really nice tree, whereas the animals don't. It's a little bit ironic given the fact that historically nobody has known how fungi are related to each other at deep levels. In fact, fungi have always been a problem phylogenetically. There are many fungi, for example, penicillin, where they had no idea what the reproductive body of that fungus was. Or there's many fungi where they had no idea even what it was and where to place it at all in the entire fungal tree of life. But we've always had ideas about animals, how they're related to each other, even if they were wrong. But at least there's a long history of people coming up with hypotheses about how animals are related. So that's what's ironic, is that the fungal tree, which nobody really had a very good idea how fungi are related to each other, comes out so well resolved. Everything's perfect. You get this great tree of life for fungi, but animals, you throw everything you've got at it, and you still come up with an answer that's not satisfactory to systematists, is what I'd say. So here's something I just discovered recently. So July of 2007, there's a paper that's published in Science, and I missed it until I was trying to prepare for this talk, and I was sort of just saying, what's known about animal evolution since 2005? Is there any updates to this? And I found this paper. Here's the title, and this is why I missed it. The title of the paper is But a Cnidarian Worm. And this is an important paper. I mean, but I missed it because what this paper says is there is, in fact, and this is sort of interesting, but it's not as globally interesting as I found out it was. Cnidarian, remember, these are jellyfish and corals, right? This is saying there is a cnidarian worm. This is unknown until now, and it's actually, if you look at any worm, whether you're talking about a nematode worm or an earthworm, so there's worms in different phyla, but they are bilaterally symmetrical. This worm is radially symmetrical, okay, and it doesn't have a gut, has no nervous system, and it's teeny tiny, okay. So this is a weird worm we're talking about, okay. But it's not sort of like, what does this have to do with the evolution of animals? Well, it turns out in this paper they cloned out 50 different genes, and then they cloned 50 genes, and then they got a whole bunch more genes from this, and then they compared it to all other animals. So they had this enormous data set within this paper just to prove that this. Here's the jellyfish down here in purple. And this worm right here is nested within the cnidarians, the jellyfish and coral. So you got the cnidarians down here, and it's nested within there. But here's all the rest of the animals, and this tree is very well resolved. They said that all of the branches that don't have a number on it are very well supported. And this time, it's based on 129 proteins, which is 31,092 amino acids. Okay, so it's an enormous data set that they sort of embedded in this paper about a little worm. And it turns out this tree, I would say that most morphologists, paleontologists, and molecular biologists, it's a pleasing tree. When you look at it, you say, this agrees with a lot of what we think now. Our ideas have changed in terms of animal relationships, but it seems to jive pretty well. And let me just review that, because I haven't really talked about the sort of tree of life for animals. The first branch here, so the animals start right here. Okay, and if we go before the animals, these are these strange protists that I was talking about. So this is here are the chenoflagellates, the ichthyospora, these fish protists, these protists that infect fish, and they've sampled these out here, and here are the fungi. Okay, so these are the episthocons. This whole tree is, and the first animal group out are the sponges, and then if we go to the next branch. Those are the cnidarians down here, including the jellyfish and the corals. And then the next branch is a major division between protostomes and deuterostomes, sort of a major way of development. It's been recognized for a very long time historically as a major division within the animals. So that would be pleasing sort of traditional taxonomists, those who thought that they would go like this, that you have the protostomes here and the deuterostomes here. We are a deuterostome, so this is vertebrates plus echinoderms, so starfish and things like that. Okay, so that's what's included in this deuterostome clade. And then this protostome clade, there's sort of these new groups that people had recently discovered. This is from so these are the ecdysozoa, That's this clade that's labeled here in green. This is from the Berkeley Museum of Paleontology. And so some of the animals you might recognize in this ecdysozoic clade include arthropods. That includes crustaceans and all insects, so that's an enormous amount of diversity. And maybe nematodes, this is one kind of worm, rotifers, and a couple other odd phyla. So that's what the ecdysozoa is. The other major group, if I can get it to come up. Oh, darn, I didn't get it to come up. I blew it. I put the animation somehow came up in the back here. But it's the Lophotrochozoa, okay? And that other major animal group, the members of it include flatworms. It includes mollusks that I'll be talking about more, so that's cephalopods and things like that. So those are sort of major players. And it also includes the earthworms, the annelids, okay, the earthworms and leeches and things like that. Okay, so and there are morphological features that unite these, we have this large molecular tree. So it looks like maybe there is a well-resolved tree. Maybe that 2005 study didn't sample enough genes and didn't sample enough taxa to get the right story. So that's why I say, when we ask the question, we go back to this. We go back to these hypotheses. Which one of these is correct? Maybe now we're leaning towards this red hypothesis that maybe if we do get this well-resolved animal phylogeny, that maybe it wasn't a really rapid versus speciation. That in fact that the Cambrian explosion is an artifact of the fossils. So it's leaning towards that direction. But it's very recent. We're talking about 2005 suggesting this Big Bang in science, and now we go up to in 2007, and and maybe the story's changed, although they probably should have published this as a separate paper. But anyway, this is a good tree. I like this tree. I think that it pleases the most amount of people that study animal evolution, I would say. Okay, I'm going to change gears. I'm going to talk about vertebrate eye evolution now. And I think the reason I want to talk about this is that it demonstrates the imperfection of evolution. And I'm targeting sort of the highest sight that people set on this. People think that the eye is one of the vertebrate eye, and it is. It's an amazing. It's an amazing accomplishment in terms of evolution. And I want to stress this, I'm going to compare the vertebrate eye to a cephalopod eye. If we were actually to test the vision of a cephalopod versus many vertebrates, the vertebrates are going to win out, in many cases at least. But there's this one aspect of the eye I want to focus on, that's the blind spot, and the cephalopods don't have that blind spot. So evolution, and the reason for the blind spot in vertebrate and the structures are not designed from scratch, and the eye is good evidence of that. Okay, so there's Darwin's famous eye quote, and I'm going to read this eye quote because I've seen this sort of in creationist literature, or I've also seen it. I've seen it stated by people. Just this first part, and here's what Darwin said. He said, "To suppose that the eye, with all its inimitable contrivances for adjusting the focus to different distances, for admitting different amounts of light, and for the correction of spherical and chromatic aberration, could have been formed by natural selection, seems absurd in the highest degree." So people pointed to that and said, even Darwin thought the theory of natural selection was absurd. But they left out the rest of the quote, actually, because Darwin goes on to point out, in his long-winded way, that it's not so absurd. And what he said is, when it was first said that the sun stood still and the world turned around, the common sense of mankind declared the doctrine false. But the old saying of the voice of the people equals the voice of God, as every philosopher knows, cannot be trusted in science. Reason tells me that if numerous gradations from a simple and imperfect eye to one complex and perfect can be shown to exist, each grade being useful to its possessor, as is certain the case, if further the eye ever varies and the variations be inherited, he's describing what natural selection is right now, as is likely certainly the case, and if such variations should be useful to any animal under changing conditions of life, then the difficulty of believing that a perfect and complex eye could be formed by natural selection should not be considered as subversive of the theory. Okay? So that's what Darwin, if you read Darwin, he's very long-winded, but he gets to the point eventually. So what he is saying here is, when we think about if we can demonstrate these sort of intermediate steps, we can imagine that they're all useful to the possessor, then imagining that this eye, in terms of, is not so absurd. And in fact, vertebrates might not be a great example. All vertebrates have a similar structure in their eye, and their eyes are all pretty good camera eyes. That is, for most vertebrates, they are taking the image, it's passing through a lens and it's inverted on the back of the retina, and then it's interpreted in the brain. It's pretty advanced. And there are not a lot of these sort of intermediates that Darwin talked about. But when we look at mollusks, where cephalopods are, we see everything in between, all the way from a limpet sitting there with just crude light-sensing organs. So this is from the General Biology textbook, 7th edition. So this is diversity of eyes in mollusks. We see what Darwin was talking about. We see we see just about every possible example from a crude layer. We've got a limpet here, and we've sort of got nerve fibers and epithelium, and then we've got some photoreceptors. So it's about as simple as you could imagine. And then we've got something, in some cases we might have an eye cup, where we turn it around. We've got some pigmented cells and some nerve fibers. And then we have a pinhole camera, which is sort of a crude camera we see in some mollusks. And then we have a full-on camera eye. And this camera eye is if we just step back and look at the basic design of the camera eye, it's this convergent evolution between the camera eyes of these squid and the nautilus has a pin eye, but between squid and octopus that have these camera eyes and vertebrate eyes are really similar, at least in the general way that it works. The image comes through this lens, it's inverted on the back of the retina, and it's picked up by the optic nerve and interpreted by the brain. And the pressure for the pressure for squid and octopus to have eyes both comes from them being predators and prey of vertebrates. Okay, they have to be able to see as well as the prey they're going to capture. In many cases, it's fish, and they have to be able to see the sharks and other sharks and whales and other things that eat them. Okay, so that's where the pressure comes from. So let's look up close at a comparison between between this octopus and this eagle. In this case, we have the eagle eye. Again, if you actually did a test, the eagle probably went out in terms of visual acuity and everything else. But I'm just talking about the blind spot. And so if we compare a vertebrate versus a cephalopod camera eye, so these eyes are evolved independently in these two lineages. Mollusks evolved this camera eye, and vertebrates evolved them. This figure is from a review of Perspectives on Eye Evolution from Opinions in Genes and Development in 1995. If we look at the way the eyes are wired, so the light's coming in right here, we look at a vertebrate eye, and if we think about these light sensors, these photoreceptors, so these are the things that have to pick up the light signal that's coming in from the outside. We think of these photoreceptors attached to these nerves. I think you can think of it like Christmas lights. Instead of the Christmas lights emitting light, they're receiving light. And so the eye is set up so that the Christmas lights are facing the wrong way. The Christmas lights are the receptors, that is, are facing the back of the retina, and the incoming light has to come through all of the wiring. Incoming light has to come through all the wiring, and then it hits the photoreceptor. And all of this wiring, because the wiring is all on the inside of the eye, it's got to get to the brain somehow. So it has to come through one spot right here that I've circled. And this is a blind photoreceptors in here because it has to make room for those nerve endings to come through and go to the brain. And this is true in all vertebrates. When we look at the camera eye of they did it right. They've got the photoreceptors, and actually these are not homologous. These photoreceptors are really different. But the photoreceptors in this squid are facing the front, so the light reaches them. They've got the nerve endings, and there's no blind spot. Okay? So from a design perspective, at least at this level, the squid has a better designed eye than the vertebrate. There's no blind spot. Now, vertebrates have come up with lots of ways to get around having a blind spot, especially us with binocular vision. You've got two eyes, you don't have a blind spot. But just in this one respect, though, we seem to have a less well-designed eye, if you're going to use that word design, than a squid or a... So why is that? Why is it that we can't just turn these around? And the answer to that is a complicated one. I want to just give you a taste of that. And it involves that when you look at the development of the eye, and you compare the development of the eye in a vertebrate, and you compare that to a cephalopod, you see something pretty different. If we look at a cephalopod, the eye is going to develop all from this external, the ectoderm, so the external layer of cells that are on the outside of the cephalopod directly evolve into an eye. So you get this loops around, and you get this development of this eye, and then you get the nerves developing off of that. So the fact that you start with this external cell layer that develops directly into an eye in a cephalopod allows them to have this direct connection back to the brain of their optic nerve. Whereas in vertebrates, the eye is developing from the forebrain. Okay, so it's reliant. It's going to be, so the forebrain is attached to the rest of the forebrain, and so we have this complicated a series of events that are occurring in vertebrates involving the forebrain, looping around, and then inverting eventually. And so this series of developments that occur in the forebrain, not in the external cell layer like it is in cephalopods, forces the nerve endings to evolve on the inside of the retina, and then forces them to go through this blind spot, go through this hole in the back of the eye. And development, you would have to change this development so radically that it can't happen in vertebrates. And remember, we're starting from, in both cases in cephalopods and vertebrates, we're presumably starting from something with a very simple eye. And just the fact that to go to the more complicated eye, we involve the brain in vertebrates, but we didn't in cephalopods, and the development of the eye itself accounts for this difference in terms of the blind spot, in terms of design. So the point is, in terms of eye evolution, that evolution isn't perfect, but we really do a great job with what we have. And when we see things evolve independently, we basically can have proof that under different circumstances, things can happen differently. All right, shift gears again. I'm going to talk about mammalian orders. So before 1990, or maybe I don't know what the right date was, but there's a series of papers that appeared before 1990. It was assumed that I'm talking about the major groups of mammals. Again, I'll discuss what some of them might be, but it was assumed that these mammalian orders arose suddenly in a rapid burst of speciation at the end of the Cretaceous. That this meteor that hit and wiped out the dinosaurs opened up the door for the evolution of mammals, and that's still true. But the assumption is that we just don't know how the different orders know it, because they all evolved at once really fast, right? We didn't have a good phylogenetic hypothesis for the relationship of the orders of mammals, with some exceptions. This assumption that there was this star phylogeny, this burst of speciation, is so common that sometimes it's still erroneously done. 2005. There are three, this is placental mammal diversification at the Cretaceous, and this summarizes sort of the three hypotheses that have been, this is the traditional hypothesis. Here's the boundary orders, and then in rest of mammals, okay? So we've got the explosion, and then after that, we have this super rapid burst of all the orders. Remember, there's more than 3 orders, a whole bunch of them, but this is just simplifying for the model. You've got all these orders of mammals diversifying basically simultaneously. The long fuse model says that the Cretaceous tertiary boundary was very important, but it was important for the explosion of speciation that occurred within the orders. So the orders were already established. So in this case, there's a bunch of mammal groups in low numbers in different places, lying in different spots and lying around, that didn't really leave a fossil record. So this is a case where we don't have good fossil evidence for anything except the explosive model. So that's why, after the dinosaurs go away, then we start finding all of these mammalian orders. So that's why the story came up. But this long fuse model suggests that yes, the Cretaceous tertiary boundary was important, but it just caused explosion of speciation in numbers of mammals after the dinosaurs went away, but that the existing diversity to a large extent was already there. And then finally, the short fuse model. This is sort of that the Cretaceous tertiary boundary didn't have that much to do either with diversification within the orders or between the orders. Okay. So some of the major mammalian orders include, and these ones you might think about. So the Edentata includes rabbits. The rodent shrews includes pigs, deer, and antelope, as opposed to perissodactyls, which are the odd-toed ungulates like horses and rhinos. And then we've got elephants. Okay, so those are just give you an idea, and there's even more, a lot more mammalian orders.